This unit, we will be studying blood and the cardiovascular system. You will notice two links to the textbook for these units on Canvas. However, the first two notes videos you will watch for this unit are straight from the textbook, so you'll only need to read two sections from that link. I will go over this information in class, so make sure you're paying attention. To get started, we need a brief overview of blood circulation, which is initiated by the pumping action of the heart. Blood exits the heart via arteries, which branch repeatedly until they become tiny capillaries. By diffusing across the capillary walls, oxygen and nutrients leave the blood and enter the body tissues, and carbon dioxide and waste move from the tissues to the bloodstream. As oxygen-deficient blood leaves the capillary beds, it flows into veins, which return it to the heart. The returning blood then flows from the heart to the lungs, where it picks up oxygen and then returns to the heart to be pumped throughout the body once again. The flow chart pictured here gives a brief overview of the topics we will cover in this chapter, but there are many details that await, so let's get started. Blood performs a number of functions, all concerned in one way or another with the transport of substances, regulating blood levels of particular substances, or protecting the body. Take a minute to write down some of the functions you already know that blood performs. Then compare it to the functions I discuss here. Transport functions of the blood include delivering oxygen from the lungs and nutrients from the digestive tract to all body cells, transporting metabolic waste products from cells to elimination sites, to the lungs to eliminate carbon dioxide, and to the kidneys to dispose of nitrogenous wastes in urine, and transporting hormones from the endocrine organs to their target organs. Regulatory functions of the blood include maintaining appropriate body temperature by absorbing and distributing heat throughout the body and to skin surface to encourage heat loss, maintaining normal pH in body tissues. Many blood proteins and other blood-borne solutes act as buffers to prevent excessive or abrupt changes in blood pH that could jeopardize normal cell activities. Blood also acts as the reservoir for the body's alkaline reserve of bicarbonate ions. It also is responsible for maintaining adequate fluid volume in the circulatory system. Blood proteins prevent excessive fluid loss from the bloodstream into the tissue spaces. As a result, the fluid volume in the blood vessels remains ample to support efficient blood circulation to all parts of the body. Protective functions of the blood include preventing blood loss, because when a blood vessel is damaged, platelets and plasma proteins initiate clot formation, halting blood loss. Blood is also responsible for preventing infection. Drifting along in the blood are antibodies, complement proteins, and white blood cells, all of which help defend the body against foreign invaders such as bacteria and viruses. Blood is the only fluid tissue in the body and is classified as a specialized connective tissue in which living blood cells, called the formed elements, are suspended in a non-living fluid matrix called plasma. Blood does not have the collagen and elastic fibers we discussed of other connective tissues, but fibrous proteins that are dissolved do become visible as fibrin strands during blood clotting. If we spin a sample of blood in a centrifuge, centrifugal forces packs down the heavier formed elements and the less dense plasma remains at the top. Most of the reddish mass at the bottom of the tube is erythrocytes, the red blood cells that transport oxygen. A thin, whitish layer called the buffy coat is present at the erythrocyte plasma junction. This layer contains leukocytes, the white blood cells that act in various ways to protect the body, and platelets that are cell fragments that help stop bleeding. Erythrocytes normally constitute about 45% of the total volume of a blood sample, a percentage known as the hematocrit. Normal hematocrit values vary. In healthy males, the norm is 47%. In females, it is about 42%. Leukocytes and platelets contribute to less than 1% of blood volume. Plasma makes up most of the remaining 55% of whole blood. Blood is a sticky, opaque fluid with a characteristic metallic taste. As children, we discover its saltiness the first time we stick a cut finger into our mouth. Depending on the amount of oxygen it is carrying, the color of blood varies from scarlet when oxygen-rich to dark red when oxygen-poor. It is slightly alkaline or basic, with a pH between 7.35 and 7.45. Blood is more dense than water and about 5 times more viscous or thick, largely because of its formed elements. Think of water versus warmed maple syrup. They're not going to be able to move at the same rate because of their viscosity or thickness. Erythrocytes are the major factor contributing to blood viscosity. Women typically have a lower red blood cell count than men, about 4.2 to 5.4 million cells per microliter, versus 4.7 to 6.1 million cells per microliter in men. When the number of red blood cells increases beyond the normal range, blood becomes more viscous and flows more slowly. Similarly, as the number of red blood cells drops below the lower end of the range, the blood thins and flows more rapidly. Your blood accounts for approximately 8% of your body weight. Its average volume in a healthy adult is about 5 to 6 liters in males and about 4 to 5 liters in healthy females. Blood plasma is a straw-colored sticky fluid. Although it is about 90% water, it contains over 100 different dissolved solutes, including nutrients, gases, hormones, wastes, and other products of cell activity, proteins, and inorganic ions, or what we will learn are electrolytes. The electrolytes, such as sodium, chloride, potassium, etc., vastly outnumber the other solutes. The table here summarizes the major plasma components and can be found on page 637 of your textbook. Except for hormones and gamma globulins, most plasma proteins are produced by the liver. Plasma proteins serve a variety of functions, but they are not normally taken up by cells to be used as fuels or metabolic nutrients, as are most other organic solutes, such as glucose, fatty acids, and amino acids. Albumin accounts for around 60% of plasma protein. It acts as a carrier to shuttle certain molecules through the circulation, is an important blood buffer, and is the major blood protein contributing to the plasma osmotic pressure, or the pressure that helps keep the water in the bloodstream. The composition of plasma varies continuously as cells remove or add substances to the blood. However, assuming a healthy diet, plasma composition is kept relatively constant by various homeostatic mechanisms. For example, when blood protein levels drop undesirably, the liver makes more proteins. When the blood starts to become too acidic, a condition cleverly called acidosis, both the lungs and the kidneys are called into action to restore plasma's normal, slightly alkaline pH. Body organs make dozens of adjustments day in and day out to maintain the many plasma solutes at life-sustaining levels. The formed elements of blood, erythrocytes, leukocytes, and platelets, have some unusual features. In fact, 2 of the 3 are not even true cells. Erythrocytes have no nuclei or organelles, and platelets are just cell fragments. Only leukocytes are complete cells. Most types of formed elements survived in the bloodstream for only a few days, and most blood cells do not divide. Instead, stem cells divide continuously in red bone marrow to replace them. If you examine a stain smear of human blood under the light microscope, you will see disc disc-shaped red blood cells, a variety of stained spherical white blood cells, and some scattered platelets that look like debris. Erythrocytes vastly outnumber the other types of formed elements. This table, found on page 647 of your textbook, summarizes the important characteristics of the formed elements. We'll now take a closer look at erythrocytes and cover leukocytes and platelets in our next notes section. Erythrocytes, or red blood cells, or as you'll see me type them out, RBCs, are small cells, about 7.5 micrometers in diameter. They are shaped like biconcave discs, or flattened discs with depressed centers. Under the microscope, they appear lighter in color at their thin centers than at their edges, making them look like miniature donuts. Mature erythrocytes are bound by a plasma membrane, but lack a nucleus and have essentially no organelles. In fact, they're little more than bags of hemoglobin, the RBC protein that functions in gas transport. Other proteins are present, such as antioxidant enzymes that rid the body of harmful radicals, but most function as structural proteins, allowing the red blood cell to deform and then spring back into shape. The erythrocyte is a great example of complementarity of structure and function. It picks up oxygen in the capillaries of the lungs and releases it to tissue cells across other capillaries throughout the body. It also transports some 20% of the carbon dioxide released by tissue cells back to the lungs. There are three very important structural characteristics that contribute to erythrocyte gas transport functions. The first is its small size and shape that provide a huge surface area relative to volume, about 30% more surface area than comparable spherical cells. The disc shape is ideally suited for gas exchange because no point within the cytoplasm is far from the surface. The second is that, discounting water content, an erythrocyte is over 97% hemoglobin, the molecule that binds to and transports respiratory gases. And finally, because erythrocytes lack mitochondria and generate ATP by anaerobic mechanisms, they do not consume any of the oxygen they carry, making them very efficient oxygen transporters. Erythrocytes are completely dedicated to their job of transporting respiratory gases, oxygen and carbon dioxide. Hemoglobin, the protein that makes red blood cells red, binds easily and reversibly with oxygen, and most oxygen carried in blood is bound to this protein. Hemoglobin is made up of the red heme pigment bound to the protein globin. Globin consists of 4 polypeptide chains, 2 alpha and 2 beta chains, each binding a ring-like heme group. Each heme group bears an atom of iron set like a jewel in its center. A hemoglobin molecule can therefore transport 4 molecules of oxygen because each iron atom can combine reversibly with 1 molecule of oxygen. A single red blood cell contains about 250 million hemoglobin molecules, so each of these tiny cells can scoop up about 1 billion molecules of oxygen. The fact that hemoglobin is contained in erythrocytes rather than existing free in plasma prevents it from, 1, breaking into fragments that would leak out of the bloodstream through the porous capillary walls, and 2, from making blood more viscous and raising osmotic pressure. Oxygen loading occurs in the lungs and travels from the lungs to tissue cells. As oxygen-deficient blood moves through the lungs, oxygen diffuses from the air sacs of the lungs into the blood and then into the erythrocytes where it binds to hemoglobin. When oxygen binds to iron, the hemoglobin, now called oxyhemoglobin, assumes a new 3-dimensional shape and becomes ruby red. In body tissues, the process is reversed. Oxygen detaches from iron. Hemoglobin resumes its former shape, and the resulting deoxyhemoglobin becomes dark red. The released oxygen diffuses from the blood into the tissue fluid and then into tissue cells. About 20% of the carbon dioxide transported in the blood combines with hemoglobin, but it binds to globin's amino acids rather than to the heme group. This formation of carbaminohemoglobin, occurs more readily when hemoglobin is not bound to oxygen. Carbon dioxide loading occurs in the tissues and travels from the tissues to the lungs, where carbon dioxide is then eliminated from the body. Blood cell formation is referred to as hematopoiesis. Hematopoiesis occurs in the red bone marrow, which is a soft network of reticular connective tissue bordering on wide blood capillaries called the blood sinusoids. Within this network are immature blood cells, macrophages, fat cells, and reticular cells, which secrete the connective tissue fibers. In adults, red marrow is mostly found in the bones of the axial skeleton and girdles, and in the proximal epiphyses of the humerus and femur. The production of each type of blood cell changes in response to changing body needs and regulatory factors. As blood cells mature, they migrate through the thin walls of the sinusoids to enter the bloodstream. On average, the marrow turns out an ounce of new blood, containing 100 billion new cells every day. The various formed elements have different functions, but there are similarities in their life histories. All arise from the hematopoietic stem cell, sometimes called the hemocystoblast. These undifferentiated precursor cells reside in the red bone marrow. However, the maturation pathways of the various formed elements differ, and once a cell is committed to a specific blood cell pathway, it cannot change. This commitment is signaled by the appearance of membrane surface receptors that respond to specific hormones or growth factors, which in turn push the cell toward further specialization. Erythrocyte production, or erythropoiesis, begins when a hematopoietic stem cell descendant, called a myeloid stem cell, transforms into a proerythroblast. Proerythroblasts, in turn, give rise to basophilic erythroblasts that produce huge numbers of ribosomes. During these first two phases, the cells divide many times. Hemoglobin is synthesized, and iron accumulates as basophilic erythroblast transforms into a polychromatic erythroblast, and then an orthochromatic erythroblast. The color of the cell cytoplasm changes as the blue staining ribosomes become masked by the pink color of hemoglobin. When an orthochromatic erythroblast has accumulated almost all of its hemoglobin, it ejects most of its organelles. Additionally, its nucleus degenerates and is pinched off, allowing the cell to collapse inward and eventually assume the biconcave shape. The result is a reticulocyte, named because it still contains a small reticulum or network of clumped ribosomes. The entire process from the hematopoietic stem cell to reticulocyte takes about 15 days. The reticulocytes, filled almost to bursting with hemoglobin, enter the bloodstream and begin their task of oxygen transport. Usually, they become fully mature erythrocytes within 2 days of release, as their ribosomes are degraded by intracellular enzymes. The number of circulating erythrocytes in a given individual is remarkably constant and reflects a balance between red blood cell production and destruction. This balance is important because having too few erythrocytes leads to tissue hypoxia, or oxygen deprivation, whereas having too many makes the blood undesirably viscous. To ensure that the number of erythrocytes in blood remains within the homeostatic range, new cells are produced at the incredibly rapid rate of more than 2 million per second in healthy people. This process is controlled hormonally and depends on adequate supplies of iron, amino acids, and certain B vitamins. The number of circulating erythrocytes in a given individual is remarkably constant, as stated. However, when this gets thrown off, erythropoietin, a glycoprotein hormone, stimulates the formation of erythrocytes. Normally, a small amount of EPO circulates in the blood at all times and sustains the red blood cell production at a basal rate. The kidneys play a major role in EPO production, although the liver also produces some. When certain kidney cells become hypoxic or oxygen deficient, oxygen-sensitive enzymes are unable to carry out their normal functions of degrading an intracellular signaling molecule called hypoxia-inducible factor, or HIF. As HIF accumulates, it accelerates the synthesis and release of erythropoietin. The drop in normal blood oxygen levels that trigger EPO formation can result from a various number of things. The first is reduced numbers of red blood cells due to hemorrhage or bleeding. The second is excessive red blood cell destruction. The third could be insufficient hemoglobin per red blood cell, as if you had an iron deficiency. And finally, you could have a reduced availability of oxygen, as might occur at high altitudes or during pneumonia. On the other hand, too many erythrocytes or excessive oxygen in the bloodstream depresses erythropoietin production. Note that it is not the number of erythrocytes in the blood that controls the rate of erythropoiesis. Instead, control is based on their ability to transport enough oxygen to meet tissue demands. Blood-borne erythropoietin stimulates red marrow cells that are already committed to becoming erythrocytes, causing them to mature more rapidly. 2 to 3 days after erythropoietin levels rise in the blood, the rate of reticulocyte release and the reticulocyte count rise remarkably. Notice that hypoxia does not activate the bone marrow directly. Instead, it stimulates the kidneys, which in turn provide the hormonal stimulus that activates the bone marrow. The male's sex hormone testosterone also enhances the kidneys' production of EPO. Because female sex hormones do not have similar stimulatory effects, testosterone may at least be partially responsible for the higher red blood cell counts and hemoglobin levels seen in males. The raw materials required for erythropoiesis include the usual nutrients and structural materials, amino acids, lipids, and carbohydrates. Iron is absolutely essential for hemoglobin synthesis. Iron is available from the diet, and intestinal cells can precisely control its absorption into the bloodstream in response to changing body stores of iron. Approximately 65% of the body's iron supply, which is about 4,000 milligrams, is in hemoglobin. Most of the remainder is stored in the liver, spleen, and to a much lesser extent, bone marrow. Free iron ions, such as Fe2+ and Fe3+, are toxic to the body, so iron is stored inside cells as protein-iron complexes such as ferritin. In blood, iron is transported loosely bound to a transport protein called transferrin, and developing erythrocytes take up iron as needed to form hemoglobin. Small amounts of iron are lost each day in feces, urine, and perspiration. The average daily loss of iron is about 1.7 milligram in women and about 0.9 milligrams in men. In women, the menstrual flow accounts for the additional losses. 2 B-complex vitamins, vitamin B12 and folic acid, are necessary for normal DNA synthesis. Even slight deficits jeopardize the rapidly dividing cell populations, such as developing erythrocytes. Red blood cells have a useful lifespan of 100 to 120 days. Their anucleate condition carries with it some important limitations. Red blood cells are unable to synthesize new proteins, grow, or divide. Erythrocytes become old as they lose their flexibility, become increasingly rigid and fragile, and their hemoglobin begins to degenerate. They become trapped and fragment in smaller circulatory channels, particularly in those of the spleen. For this reason, the spleen is sometimes called the red blood cell graveyard. Macrophages engulf and destroy dying erythrocytes. The heme of their hemoglobin is split off from the globin molecule. Its core of iron is salvaged, bound to protein, such as ferritin, and stored for reuse. The balance of the heme group is degraded to bilirubin, a yellow pigment that is released to the blood and binds to albumin for transport. Liver cells pick up this bilirubin and in turn secrete it in bile into the intestine, where it is metabolized to urobilinogen. Most of this degraded pigment leaves the body then in feces as a brown pigment called stercobilin. The protein globin part of hemoglobin is metabolized or broken down to amino acids, which are released into the circulation. This concludes our notes section for today. Next time we will cover the other formed elements found in blood, leukocytes and platelets, as well as the clotting mechanism we are all vaguely familiar with after childhood.